So if you think that evolution is in fact a complicated system, and you accept that all of the cells that we currently know are very complicated, and we can't fully understand them yet, at least, perhaps in the future, hopefully in the future we will. Next slide. Then we begin to think that perhaps there's a limit to what we can learn from these sorts of experiments that the majority of the scientific community are carrying out. Next slide. And perhaps a solution would be to go from the other direction. Maybe if we build a cell or try to build a cell from its pieces rather than starting from existing organisms, then maybe we'll discover that life is simpler than we think, and maybe we'll be able to build a fully understandable system. Next slide. So again, there are many ways you can look at a cell, and if you're trying to achieve a particular life-like function, there are many different ways that you can define that. What I will mostly be talking about for the rest of my time is the first one, replication. There are a lot of obvious criticisms for why replication is not an excellent definition of life. And in fact, my laboratory does work on other aspects of trying to build cell-like function. But I think this is the easiest to address, and certainly the one that we have the most data on. And why? Why this focus on replication? Well, it goes back, next slide, to the same thing that I had been mentioning, which is evolution. In order to have evolution, you must have a replicating system. And sort of, I think the dream for many of us is once we build a simplified replicating system, which requires design. We're designing and building this initial system. Once we do that, then we may not necessarily need to design further processes. Maybe then we can have the system evolve towards what we want. So really, I think the dream is to build a self-replicating system capable of evolution that we can then evolve to whatever function that we want. Next slide. But there are many different ways you can envision doing this. You can envision starting from a pure chemical perspective and building something just based on chemistry that displays the properties of life. Or you could envision reducing natural organisms, as I discussed. Or you could think of going to several different living organisms and looking at the simplest ways they achieve each function and put those together. So for example, maybe one cell is known to require 10 proteins to copy its genome, whereas another organism may require 20 proteins to copy its genome. Well, then we would take the one that only requires 10. We would use that system and then look for other processes that we need from other organisms and take the simplest system from each one. I often refer to this as sort of a hybrid approach. So first I'll show some slides on the chemical approach to trying to build systems that look lifelike, and then I will try to show a couple of slides on this hybrid approach. So if you want to build, next slide, a cell-like system, and you're focused on this concept of replication, then you can break this down into 4 individual steps. You would probably need a compartment, so essentially a lipid membrane to house your living system, and it provides it with identity. You would want this compartment to grow, which I'm showing as step 1. You would want this compartment to be able to feed and to release waste, that's shown as step 2. You would want this compartment to have inside of it a genome, and you'd want that genome to be able to replicate. I'm showing that as step 3. And then you would want the compartment to divide, which I'm showing as step 4. So you can break down this process of replication into 4 steps. Next slide, please. And the amazing thing is you can get a lot of these things to occur without using any biological machinery. You can use just simple chemical physical forces. And this is particularly true if you begin to look away from the types of molecules that are used in biology. So if you use the kind of lipids that are exploited in biological cells, you need lots of proteins encoded by genes in order to get the compartment to grow. But if you use more simple lipids, these simple lipids will spontaneously form compartments, and if they are mixed together in the right way, they will spontaneously grow. So you don't need any proteins, you don't need any genes. These are vesicle systems that can grow on their own. Next slide. So that was growth. I'm obviously making things very simplistic, and I'm putting just about 1 slide on each process. So I'm happy to answer questions. I'm not showing all of the details. So if the next step is nutrient exchange, how do we deal with this problem? Next slide. So in biology, again, in order to get specific molecules into the cell and release specific waste molecules, we have highly complex protein machinery to carry out those tasks. But with the same simple lipids, these lipids have an inherent selectivity, where they form membranes that are inherently selective. And this probably has to do with the dynamics of the lipid systems that we are using. So they're a little bit more dynamic. They move a little bit a lot faster. They move around a lot faster than the kind of lipids that you find in a biological cell. And we think it's this sort of dynamics that give the system these permeability characteristics that allow it to accept material and to release material. And it can do it specifically. So some molecules go through the membrane extremely fast, in the order of milliseconds, and some molecules take hours, and other molecules don't go through at all. And many of the molecules that go through easily are molecules that you find in biological cells. So for example, ribose, which is a simple 5-carbon sugar, which we use for energy in the form of ATP, which we use as part of our genome. Our genome consists of ribose units. Ribose goes through our membranes extremely fast, on the order of seconds, whereas other molecules that are a little bit bigger, or some that are even the same size but are not ribose, go through significantly slower. So you can begin to see how you can build systems just taking advantage of the intrinsic characteristics of the molecules themselves without trying to build in specific genetic or protein machinery. Next slide. The next step that I'm going to be talking about, I'll actually skip genomic replication just for a second. I'll come back to it in a minute. But the next one I'll talk about is compartment division. So next slide. And this, surprisingly, was done by Tim Zhu in the Shostak lab. This is not work that I carried out. But what he found, interestingly, was if instead of making membranes that only have one layer of lipids, you make membranes that have many layers of lipids, like an onion-like structure, which is, in fact, easier to do in the laboratory, then as these systems naturally grow, they also naturally divide. This is through chemical and physical processes that are not fully understood right now. We have some hints as to why, but even if we don't fully understand why it's happening, it is something that we can actually see under the microscope with our own eyes. I mean, it's really happening. If you have these sort of onion-like shaped compartments in a test tube and you allow them to grow, they naturally divide. Next slide. And so the final step of building a chemical cell is genomic replication. Next slide. And here we had to use a little bit of organic chemical tricks. The kind of building blocks for building DNA in a cell do not spontaneously polymerize, they do not spontaneously copy. They require enzymes to carry out these processes. But you can modify the molecules a little bit and you can make them more reactive. And so if we do this, if we use a few chemical tricks to copy, to create a system that can copy DNA, next slide please, then we can start putting all of these things together. And so here I'm showing just a little schematic of 4 molecules. I mean, this is really, really basic. I'm not talking about 4,000 genes in E. coli. I'm talking about 4 chemicals. The first one, the fatty acid, which is the very simple lipid molecule I've been talking about on several of these slides. 2 pieces of DNA, which I call a primer and a template. And then these chemically modified nucleotides, these activated nucleotides, the building blocks for the DNA. If we mix these components together, next slide, then we can actually build a system in which we have vesicles that feed, so they uptake these activated building blocks for DNA. So they feed nutrients, and they use that nutrient to copy DNA that it already has inside of it. So this is a feeding and replicating system. This is not life. There's no way I would ever try to convince you that this is alive. This does not possess all of the characteristics of one full replication cycle. There are many criticisms you can make, but it is, I think, quite impressive that with 4 chemicals, we have a system that is beginning to show replication ability. So this is very simple chemical system. Next step, please. And in fact, for this replication process, each of the individual steps that I've talked about have been done in the laboratory. We can do each one of these steps. And nobody has taken each of these individual steps now and made a full cycle in which you build a system that is just constantly replicating its genome and growing and dividing. We can do each step individually, but there's still a little bit more work that's needed in order to make the system fully functional on its own. Next slide. But I think it's exciting. I think with just a small number of chemicals, we're beginning to show that you can build emergent lifelike properties. So I think it shows that it is possible to build lifelike systems from the bottom up, and the system could potentially be evolvable. That's next slide. I do want to emphasize that there are a lot of limitations. We haven't built the full replication cycle yet. Our system is only stable under really narrow solution conditions. You really have to have exactly the right conditions in the laboratory for the system to work. It's not robust in any way. And there are a lot of other criticisms one can make, which of course are true. This is just the beginning stages of our work. Next slide. So what if we want to build with biological instead of just using simple chemistry?
We're running a bit out of time. How are you doing for time? How are you doing in terms of your total talk?
I have probably about 6 or 7 more slides, but I can stop now if you want me to.
Well, not if you still have a very crucial point to make.
Okay, I will just show a couple of slides and then I will stop. So I won't. Is that okay?
Yes, great, please.
Okay. So you can also imagine, let's go to the next slide, building what I had talked about before, which can you take the individual pieces of life and put them together inside of a compartment. And so this is something that we are trying to do now. It's something that has not been done before. And let me see how many slides we should skip ahead. I'll just show one of the pretty slides. 1, 2, 3, 4. If you could go 5 slides ahead. More? Okay. So that's an example of us taking machinery that is used in a cell to divide a cell. So in a cell, it takes about 9 to 15 proteins to divide a bacterium, which is, what is assumed. It's not really known as a fact. We've taken just one of these proteins and we've started to try to build a division system in the laboratory. And here you can see that these molecules are aggregating inside of the compartment. They are not dividing the compartment. This is really just step 1. But you can see that we're starting to build the machinery. And if you go to the next slide, so there was too many ahead. Okay, never mind. Let's go back to the previous one. Okay, so the one before it was the one I thought we were showing. Okay, so no, go forward one. Forward, yes, this one. No. So we can also put the synthetic DNA inside that encodes this division protein. And I know that it's not very clear, the image is not very clear, but if you look carefully, you can begin to see on the edges of some of these compartments some deformations. So with just a single protein, I think we're starting to build a system that should be able to divide a compartment. And I'm happy to stop there. You can just go to the last slide so that I can show the people who worked on this.
So, am I in your picture now?
Yes, I am.
Thank you very much, Sheref.